Corals and Light is a project located in the marine biology section in Elsinore. The project has several implications. One of them is understanding how organisms use light and how efficient they are in creating energy. Daniel brings the coral to the lab carefully.
It's a nice day.
Daniel has worked with OCT technology for several years.
What we can see here is a coral, a fluorescent coral, in seawater, and it's basically connected to this OCT system. So that's an imaging technique that allows us to look inside the tissue. That's one polyp here, and that's the opening, the oval mouth. And all the green things here by the side, they're called fluorescent pigments. And we're interested in the function of these pigments. With regard to studying corals, it's basically they're beautiful, and they're fascinating, and we don't understand a lot about them. They're basically these weird creatures living underwater and occurring in all these different colors, and they're some kind of mix between an animal and a plant, and they're very fascinating. We can get detailed structural information not only on the surface but also within the tissue. Because with OCT we're able to image the coral tissue live. So we can look at these behavioral modifications. We can look at changes in tissue structure. Now what we can see here now is an OCT scan, 3D, and you know the different colors here represent different intensities of the signal. You can see the two tissue layers here. So that's the upper tissue layer, and then there's the second tissue layer. And with that OCT, we can scan through it. So we can scan through the coral. And we don't have to prepare the tissue, and the coral is alive. We can just look inside and see what is happening there. When there is dim light, then the tissue will expand, and these pigments are more loosely incorporated into the tissue, which then leads to strong light scattering and actually enhancing photosynthesis. And so it's this kind of dynamic modulation that a coral can do, that potentially can do, that we are testing with OCT, because with OCT we're able to image the coral tissue. Now the special thing about this coral is that it's a fluorescent coral. What that means is basically, if we make this dark here for a second, and now we shine blue light onto it, you can see how the fluorescence pops out. So that's basically caused by these, they're called green fluorescent pigments, that absorb light in the blue and then re-emit it at a higher wavelength, which is green in this case. The challenge with understanding photosynthesis in these organisms is that they have complex tissues that modulate the light environment strongly. So we're trying to understand the optics of these tissues in relation to photosynthesis.
It is a highly interdisciplinary project involving different scientific disciplines such as biology, physics and chemistry. Daniel returns the coral to the aquarium.